Hi, this is Michael and Junsu, and we'll be going over the acetylcholine signaling pathway. Introduction. Acetylcholine is a key neurotransmitter involved in memory, muscle contraction, and organ function. Acetylcholine is also a part of the autonomic nervous system, which regulates smooth muscles, blood vessels, bodily secretions, and heart rate. Acetylcholine is also part of the parasympathetic nervous system, which helps maintain homeostasis by slowing heart rate and increasing digestion. The acetylcholine receptor is a G-coupled receptor.
Alright, so I'm gonna be going over some general information about the neurotransmitter acetylcholine. So, acetylcholine is a polar molecule and is thus hydrophilic. Since it is polar, the binding site of acetylcholine must be outside of the cell or the muscle fiber membrane. And then here's a brief overview of how acetylcholine or ACH works. So first, a nerve signal or an action potential reaches the axon terminal. This causes calcium ions to enter, which triggers the release of acetylcholine into the synaptic cleft. Acetylcholine then binds to nicotinic receptors, which leads to muscle contraction, heart rate regulation, and secretion by the cell. This signal is stopped or terminated when ACH is broken down by the enzyme acetylcholinesterase or ACHE. Okay, so here's a more detailed explanation of how the acetylcholine signaling pathway works. So these two red shapes are the nicotinic acetylcholine receptors, and these yellow highlighted portions, those are the binding sites of the receptors. So the first step of the process is an action potential arrives. So an action potential is going to travel down the axon and go to the axon terminal. So this is going to be an action potential, or otherwise known as a nerve impulse. And then what this does is this is going to cause the voltage gated calcium channels to open, and this causes calcium ions or Ca2 plus to travel from outside of the axon terminal into it because it's just flowing from high to low concentration. And then the influx of calcium ions causes acetylcholine. This is going to cause acetylcholine to travel from the axon terminal, and it's going to cause it to travel to these like receptor proteins and bind to the binding site. So acetylcholine is going to be released into the synaptic cleft. So acetylcholine or ACH. Alright, so next, once the acetylcholine binds to its binding site, you can see that that's denoted by like the red stars on the binding sites. The receptors like move apart, so you can see like they've moved apart and there's a gap in the middle, and this is going to allow ions to flow through it. So inside the muscle fiber membrane there's going to be potassium ions, so K plus. I'm going to draw that on like the inside of the cell membrane, and then on the outside there's going to be sodium ions, so like Na plus on the outside. And this leads to, once the ion channels open, this leads to rapid depolarization because the Na plus are going to move across. They're going to go into the muscle fiber membrane as they move like down its gradient. And some of like the leftover Ca2 plus, some of the leftover ones are also going to move into the membrane as well. So this causes rapid depolarization, as I said, and this leads to muscle contraction until termination of the signal. So this keeps going until like the acetylcholine leaves the binding site. Alright, so finally, once the signal has been transmitted, the acetylcholine that's attached to the binding site, it needs to be removed to terminate the signal. So what happens is like these acetylcholines that are attached to the binding site of the nicotinic acetylcholine receptors. These are going to bind with an enzyme called acetylcholinesterase, also known as ACHE. So they're going to like come here and attach to the enzyme. So this enzyme it's just present in the somatic cleft, and what it does is it breaks down the acetylcholine into two components that are inactive. So it's going to break it down into acetate and choline. So what this does is this prevents continuous stimulation of the a cell, and because if the ACH were like permanently bound to the receptor, it would like cause Na ions and possibly Ca2 plus ions to like keep continuously flowing into the membrane, which could lead to like problems like muscle spasms. So once the enzyme breaks ACH down into acetate and choline, this the choline that's produced by the breakdown or the hydrolysis of ACH is taken back up like all the way to like the start of the cycle up here, and it's going to use for future signaling to produce and release more ACH to keep the process going. Here's a brief overview of the sequence of events in acetylcholine signaling pathway that I talked about.
Advantages versus disadvantages. Advantages includes nicotonic receptors, which are fast signaling and essential for movement and cognition. And also another advantage is that it modulates organ function and is used in medical treatment. However, the disadvantages are it can desensitize and could lead to addiction. And side effects include slow heart rate and gland overactivity. Acetylcholine signaling pathway, key players in the signaling pathway. Signaling molecule is acetylcholine, and for the receptors is nicotonic, and nicotonic ion channels allow Na plus and Ca2 plus influx, which leads to rapid depolarization and muscle contraction. Intended responses of acetylcholine signaling pathway. Nicotonic receptors are fast synaptic transmissions, and this leads to muscle contraction and nerve signal propagation. And muscarinic receptors are smooth muscle contraction and gland secretion. Overall functions include movement, heart function, digestion, and brain activity. Termination of acetylcholine signaling pathway. Acetylcholinesterase breaks down acetylcholine into choline and acetate, and this stops receptor activation, which ensures controlled signaling. Agonists and antagonists of acetylcholine signaling pathway. To begin, agonists are substances that activate receptors to produce a biological response. In the case of acetylcholine, we both have endogenous and exogenous agonists. Endogenous agonists acetylcholine are the natural neurotransmitter for both nicotinic and muscarinic and are receptors in acetylcholine. It plays a key role in muscle activation, autonomic nervous system function, and brain signaling. Exogenous nicotine activates nicotinic receptors, and when it binds to these receptors, it stimulates the nervous system, which leads to an increase in alertness, heart rate, and in some cases addiction due to its effects on dopamine release. And lastly, now we will be going over how agonists and antagonists affect the pathway. Agonists enhance the effects of acetylcholine by activating its receptors, and this leads to stronger nerve and muscle activation. For example, nicotine stimulates the nervous system, increasing heart rate, while muscarine enhances gland secretion and it lowers the heart rate. And on the other hand, for antagonists, it blocks acetylcholine from binding to its receptors, and this reduces nerve and muscle activation and slows organ function. Curator, for instance, it blocks nicotinic receptors, preventing muscle contraction and causing paralysis, and atropine blocks muscarinic receptors, and it increases the heart rate and reduces gland secretion. And yeah, thank you for listening, and this was Michael and Junsu's project on how about acetylcholine signaling pathway.